So with the permission of our respected speakers, I'd like to open our webinar today. Good afternoon to our Europe and Asia participants, and good morning to our US participants. I'd like to welcome everyone to today's webinar, Solving Nucleic Acid Sequencing Challenges, ClickSeq Applications in RNA and DNA Library Preparation. My name is Nasifa Hamidzada, and I'm Head of Business Development, Marketing and Sales at baseclick, and I'll be your moderator for the sessions. We are excited to bring together three leading scientists to showcase how ClickSeq, a next-generation sequencing platform based on click chemistry, is being used to overcome the key limitations in RNA, DNA library preparation, from recombination detection to polyadenylation profiling. I'll start with a short introduction to our speakers. So we'll begin with Dr. Thomas Frischmuth, CEO and CSO of baseclick GmbH. Dr. Thomas Frischmuth has over 30-year experience in life science and medical technology fields. His research activities span chemical synthesis, molecular and cell biology. Dr. Thomas Frischmuth has been also involved in development and evaluation of several drugs with one current market for cancer treatment. Today, he'll introduce the chemical foundations of ClickSeq and how this unique approach improves library preparation. Next, Dr. Andrew Routh, the Chief Scientific Officer and the co-founder of ClickSeq Technologies, as well as an institute investigator at Scripps Research. Dr. Routh will present the biological applications of ClickSeq, including viral genome sequencing and splicing analysis. And finally, we'll hear Dr. Eric Wagner, professor in the Department of Biochemistry and Biophysics at the University of Rochester. Dr. Wagner has led an NIH-funded lab for 18 years, published approximately 100 papers in RNA biology, and serves as an associate director of Center for RNA Biology and co-director of New York State Teacher Certification Examinations. He'll represent how ClickSeq is used both in transcriptomic research and in biology. Before all, I would like to just introduce the history behind the development of ClickSeq kits. As the name says, ClickSeq is an advanced method for DNA and RNA library preparation. The technology is patented by ClickSeq Technologies, and the ClickSeq kits were developed in a cooperation between baseclick and ClickSeq Technologies. Currently, baseclick GmbH is the OEM partner for the ClickSeq kits. So at this point, I'd like to give the word to our first speaker, Dr. Thomas Frischmuth. And yeah, so Thomas.
Okay, thank you very much, Nasifa, for introducing myself as well as baseclick. And I want to first initially tell you a little bit about the click chemistry, and then leave it to my co-referee reviewer to basically introduce the application of it. So the click chemistry, and in particular what we also use for the ClickSeq kits, is a click reaction. And I just like to introduce here the two different type of click reaction. The concept was introduced by Professor Barry Sharpless, who actually received 2 Nobel Prizes in his life. And he is a co-founder advisor of baseclick. And he basically, and that's, sorry, I have to get the pointer. Is there no pointer? Okay, sorry. Basically, he introduced the kappa-mediated click reaction, which is depicted here on the left side. However, there is also in the click chemistry is a non-kappa click reaction type, which is or one of these reactions is a so-called strain-promoted click reaction, which was basically from Caroline Petozzi developed. And both also by side Barry, also Meldal and Petozzi actually received the Nobel Prize for this reaction type. And as you can see, it is more or less a conjugation between an alkyne group and an azide group, which actually then gives this triazole linkage. At baseclick, we more or less developed a lot of methods and technologies using this click chemistry to conjugate nucleic acid with all kind of different either chemical moieties, but also biological moieties such as peptides, proteins, and others. This application actually are now used very widely in various applications, such in virology, RNA biology, synthetic biology, therapeutics, diagnostics, gene therapy as well, and that will be subject of today in sequencing tools. And today the part will be in sequencing tools for library preparation. However, the click chemistry is also being used and developed by baseclick also in the kit systems of Pacific Bioscience, as well as the new SBX platform of Roche. Just as an introduction, the click chemistry at a glance is, as I already mentioned, is a chemical ligation method, which actually is bioorthogonal. So you can actually connect in different ways the moieties from left to right side. And basically, you can either introduce on one side the azide and on the other side the alkyne, or reverse. And also, you can actually use that method to label various type of nucleic acids in various parts. And that is more or less shown a little bit on the pictures here, which we published in the paper, Orthogonal End Labeling of Oligonucleotides, recently in Chem Biochem in 23. And basically what we actually did here is we modified the 3' or 5' ends of oligomeric RNA or DNA, and basically showed that indeed you can actually conjugate on the physiological conditions various groups. And also, and that's important for the ClickSeq technology, you can also link oligomeric nucleic acids with other oligonucleic acids, either a SPAC reaction or in the kappa-mediated reaction. Here, I just want to point out that this technology in the last, especially last 2, 3, 4 years, were more or less also introduced into the therapeutic field. In particular, since 2017-18, we were able to more or less conjugate oligomeric RNA with all kinds of different ligands. And that's, we published in this paper, where we actually showed indeed when you actually add via click chemistry multimeric ligands or other kind of targeting sequences, you can actually specifically target the dendritic cells. And one of these molecules is now in the process to be in a phase 1 study in a microRNA therapy. And we published all this in Journal of Carbon hydrate chemistry, as well as in nature communication, basically. And also, and that actually was more or less also published recently by our group, is we were also able to introduce the click chemistry in mRNA engineering. So, and that actually shows you how sensitive this method is. Even the click chemistry is able to undergo chemical conjugation with a molecule which more or less is very sensitive to changes in like the pH or other kind of acidity and so on and so on. Basically is we were able to introduce clickable modified nucleotides, and by that, and that is actually published in this paper, we're able first of all to click conjugate these kind of groups, but also what we showed is they actually suppress immune response, lead to protein expression and especially prolonged expression. As well, you actually avoid certain RNA folding to avoid in therapeutic development the formation of hybrid RNA. Coming now to the application click chemistry in what we call in NGS, and basically is, as I already mentioned initially, is indeed we, together with the group from Andrew Routh and ClickSeq, we developed the technology they actually introduced to a kit system, which you now can actually purchase as shown by Nassifah. And basically is what actually is done here is you introduce a clickable group at the 5' end in reverse transcription. And basically at the end of the day, you can actually also, and that actually, as I already mentioned, this one is again oligomeric cDNA. You can actually click an oligo for PCR amplification onto this clickable group as it is depicted here. And this leads that you actually can replace the fragmentation, which are physically or enzymatic. And also that's more particular important is you can actually more or less do the click reaction as it is shown here for the ligation. The amplification is the same. We also have developed a special kit for long read. Basically, what you do is you do not actually stop as it is shown here. In various points, you basically leave the reaction until the end of the mRNA, 5' end of the RNA, and then do the click reaction. And just to point out, really, and that is actually the I will now actually will basically hand over to Andrew as a next speaker. Basically, this is the important point in that we actually avoid the enzymatic ligation of these fragmented pieces of cDNA or DNA, which then actually lead to a lot of false and bias and error introduction in the library. For at the end, you amplify a lot of errors, and then in the sequence, you have to do a lot of purification at the end via your software to actually analyze these kind of ligating after. Thank you very much for actually listening, and the discussion will be afterwards. And I actually like to hand over to my colleague, Andrew.
Thank you very much for the introduction, Thomas, and it's a real pleasure to be able to present ClickSeq to you. I'm going to give a brief overview on some of the applications and technologies that we've developed using ClickSeq as a platform for next-generation sequencing. Briefly, I have two hats. So together with Elizabeth Jaworski, we founded ClickSeq Technologies as a company to provide NGS kits and services. And then I'm also Institute Investigator at Scripps Research in La Jolla, where I have NIH-funded research to perform research studying RNA virus recombination studies. And so in my lab, we're really interested in this process of RNA recombination. So how essentially RNA recombination is during the replication of an RNA virus, the polymerase can jump from one template to another, essentially stitching together two new templates to form a new chimeric virus. And this is really important for the way that viruses evolve because it allows a virus to shuffle mutations or single nucleotide variants between progeny virus, generating new diverse templates as it does so. And so viruses replicate very, very quickly, and they have to balance the error-prone nature of the polymerase with recombination to allow for the formation of wild-type viruses, as well as to purge deleterious mutations or lethal mutations in the virus. And within a single cell, you might have thousands of copies, tens of thousands of copies of a single RNA template that are being replicated at any one time. And so this process of homologous recombination is actually very prolific within a single cell. And any genome that comes out of a cell, between 1 to 25%, depending on the virus you're studying and this host system, could be a product of recombination. So it's a very common process, very frequent process. And this can be particularly important when you have two viruses that happen to infect the same cell. So you have a co-infection. So you can actually get recombination between the two viruses. And there's plenty of examples of this in nature. And just throughout the pandemic and the SARS-CoV-2 pandemic, we've seen this occur multiple times. So the most recent variant, for example, XEC, was actually a recombinant between two different substrains of Omicron. And then sometimes recombination can occur between non-homologous sites. So in other words, you get a recombination event that deletes or inserts nucleotide sequences into the viral genome sequence. And this occurs frequently as well during virus replication and can result in structural variants, such as the insertion that we saw in the furin cleavage site of SARS-CoV-2, and can also result in the formation of defective genomes, or defective RNAs. And these defective RNAs occur routinely in nature, and they can affect the outcomes of disease or the efficacy of vaccines. And they can also occur in experimental settings. So if you're passaging a virus in the lab, you can have a lot of these defective RNAs building up during the passaging of the virus, and they evolve over time. And so next generation sequencing is a really excellent tool for studying diversity of RNA viruses or DNA viruses. And so as a sort of general approach, you can take a purified virus, you can take the extracted RNA, and you can subject it to the next generation sequencing. And very briefly, you would take that RNA, fragment it into lots of these short little chunks. You would ligate on or use PCR to add on sequencing adapters and then sequence those on an Illumina platform. And what this allows you to do is, because the process is essentially quantitative, each of those little chunks will tell you about the sequence diversity in the original RNA sample. So you can start to count things like point mutations or sequence variants. And importantly for our research, we can use bioinformatic tools to map recombination events that occur within our original virus sample. And so when I first started studying RNA recombination, there are actually quite a few problems with the way that next generation sequencing processes worked. So in general, the processes require RNA fragmentation. And after the RNA fragmentation, you'd ligate on sequencing adapters. And a large problem here is that sometimes you actually ligate the RNA to other RNAs of the virus template. So you're generating artifactual chimeras through these ligation steps. And this is pretty hard to avoid when using enzymes. Secondly, by having fragmentation, when you do reverse transcription, when the reverse transcriptase gets to the end of an RNA fragment, it tends to jump onto another RNA fragment in a forced copy choice mechanism, which again also generates artifactual recombination. So essentially, you're using a viral protein, reverse transcriptase, to study the process of viral recombination, which is a problem because then you have a lot of artifacts that you can't disentangle from the real signal that you're interested in. And so this is why ClickSeq ended up being a really good solution for studying RNA recombination in virus samples. Firstly, we don't have to fragment the RNA. You can just start off with total RNA straight from your biological sample. And then in a simple reverse transcription reaction, we can spike in these azido-nucleotides. So in other words, we have around a 1 to 20, 1 to 40 ratio of azido-nucleotides that have this terminating azido group with our normal deoxynucleotides. And then during the reverse transcription reaction, these azido-nucleotides will get stochastically incorporated into the cDNA. What this means is that prevents the template switching from the reverse transcription reaction. It also means you don't have to fragment your RNA. It also means that you can exploit this azido group at the end of your cDNA using click chemistry. So we can take an oligo, for example, we just take an oligo which has around 60 nucleotides for the Illumina sequencing adapter, and we can click this using copper click chemistry directly onto the cDNA that we just synthesized. This generates this rather large and unusual triazole backbone in the single-stranded cDNA, but nevertheless, you can take this click-linked cDNA and just put it straight into a PCR reaction to generate your final next-generation sequencing library. And so we tested this directly to see how this affects RNA recombination. We can take two viruses from two completely different systems, we can mix them in a pot and then subject them to ClickSeq sequencing. And when we do this, we can map the recombination events that we don't expect to find between our two different viruses to measure the rate of artifactual recombination. And we found in these situations that we only found 25 reads from around 23 million reads of artifactual recombination. So that brings the artifactual recombination rate down to around 3 chimeric reads per million mapped reads, which is considerably better than any other available technologies that we tried. And so we've used this method extensively in my lab over the years to study the mechanisms of RNA recombination in a really fine-grained, detailed way in our lab. So we've published many papers on this, and you can go to our website and look at the different resources that we have where we go through all these in details, but I just wanted to highlight 3 specific studies. So one of the first studies we published was in this virus, flockhouse virus, that readily generates defective RNAs and generates a lot of products of non-homologous recombination when you passage it in cell culture. And so we could take samples of the virus during a 1-month passaging experiment and subject each sample to ClickSeq sequencing. And we found only after 3 or 4 passages could we start to see the emergence of these RNA recombination events. So because the method was so sensitive and because we had such a low background of noise, we could really dig into the mechanisms of where these recombination events are occurring early on during passaging, and thus how they affect the evolution of defective RNAs in flockhouse virus later on during passaging. More recently, we've been looking at RNA recombination rates in coronaviruses, and what we found when we sequence using ClickSeq different coronaviruses such as SARS-CoV-2, MERS, 229E, and OC43, the recombination rate of SARS-CoV-2 is considerably higher than any of the other beta coronaviruses that we've sequenced in the lab. And this might reflect one of the reasons why we see so many recombinants and structural variants throughout the pandemic. Finally, in a paper we recently published with David Barton from University of Colorado, we're looking at the mechanisms of recombination between enteroviruses. And recombination in enteroviruses is an important feature of how we get vaccine-derived poliovirus, for example, where you have recombination between the live attenuated oral poliovirus and circulating enteroviruses that might be in the environment. And so by recapitulating that event in cell culture, with ClickSeq, we can sequence that exact junction between the circulating enteroviruses and the poliovirus subgenome RNA that we had in the experiment. But because the method is so sensitive, we can start to find these other minor recombination events as well. And they sort of occur around the same region between this P2 and P3 region of the viral genome. And that's important because it tells us there isn't just one event that's generating these recombinant genomes, there's actually a number of different events that occur with different frequencies, which reflects the sequence homology at each of these sites. And so only with a very sensitive method like ClickSeq can we start to dig into these quite refined details of where RNA recombination takes place. And so beyond RNA recombination, we can also use ClickSeq as a tool in the lab to study structural biology and the biophysics of RNA viruses. So coming back to our favorite virus, flockhouse virus, this is a really interesting tool for studying RNA virus structure in the lab, because you can take the virus particle and you can heat it very gently in the lab. And what you start to see are these puffs of RNA that start to extrude from the virus particle itself. And we think that this recapitulates the process of the RNA being released from the virus particle as it enters a cell. So we can essentially use structural biology to study the different structural intermediates during RNA virus disassembly and infection into a cell. And what's curious about these puffs is that they tend to be around the same size. They tend to extrude from the same axis on the virus particle, but we don't really know what RNA composes these puffs. Is it RNA1? Is it RNA2? Is it 3' or 5'? And we don't really know. But because with ClickSeq, we don't have to do any fragmentation, we can actually put these viruses into our sequencing reaction directly. We don't have to purify the RNA, we don't have to fragment, we don't have to do anything clever. We just put it directly into our reverse transcription reaction. And so when we do this, if there's protein blocking the RNA, you can't prime there, and you certainly can't reverse transcribe through it or incorporate the azido-nucleotide. So in other words, the only RNA you can sequence in this scenario is the exposed RNA. And we recently released a preprint where we did this with flockhouse virus, and the answer was quite clear and quite satisfying. So when we compare the exposure of RNA between these intact virus particles and these puff particles, we find that the RNA that is extruded is right at the 5' end of RNA1 and right at the 3' end of RNA1, which makes sense because this is where some of the structural motifs required for translation and replication of those viral RNAs are present. So it makes sense that those be released first into the cell so the cellular machinery can grab onto them and start to express and replicate the RNA virus. So far I've described methods for using ClickSeq in the lab as an experimental tool, but really wanted to push the boundaries further. We wanted to see, well, can we use this as a tool to start to study these features in clinical samples as well? And so this is why we developed Tiled-ClickSeq or targeted ClickSeq. And so very briefly, for example, in SARS-CoV-2, you also have recombination events that occur during a natural infection. A patient expressing SARS-CoV-2 will have recombinant genomes inside them. And so instead of using random primers in our reverse transcription reaction, we can take RNA from a patient infected with SARS-CoV-2, and we design hundreds of tiled primers strewn across the viral genome that recognize SARS-CoV-2 specifically, and use those as our input in our reverse transcription reaction in ClickSeq. And what this means is that we're going to specifically sequence our virus of interest rather than all the other stuff that might be there, like the bacterial RNA, DNA, the human RNA, the human DNA. So this is very similar to many amplicon-based sequencing approaches for enriching a virus of interest when you're doing clinical surveillance or virus genomic surveillance. But there's one important feature that makes this different. So because in ClickSeq, we're using one specific primer at one side of the amplicon, and the other end of your amplicon is generated by that stochastic incorporation of the azido-nucleotide. What this means is you can generate random fragments from your target of interest only using one primer per amplicon. And this may seem like a simple adjustment to the protocol, but it has some important consequences, because what that means is that you can essentially sequence anything that's upstream of your target of interest. So you have a recombination event, or if you have a splicing event, or if you have some sort of fusion between your virus and the host, or your virus and another virus, or even the positive sense RNA, the negative sense RNA, it doesn't matter. You don't have to have two pairs of primers, you just have one, and you can sequence anything upstream. And so this provides a really powerful and agnostic tool for surveying RNA recombination in complex clinical samples. And so during the pandemic, we received a grant from the CDC to deploy this approach to study SARS-CoV-2 in Galveston, Texas. And we started off by sequencing isolates of SARS-CoV-2. We designed around 72 primers that targeted the virus genome, which are depicted along the length of the SARS-CoV-2 genome here. There's around these little dots at the bottom. And in orange and blue is the sequence coverage over the virus genome when we sequence with either nanopore sequencing or with the Illumina platform. And what you'll see is you get these nice sort of what I call sawtooth pattern of sequencing coverage along the virus genome as we go along. Importantly, because we have multiple primers and we get lots of different reads coming off the primer at the time, those amplicons that come off will actually overlap. And because they overlap, we can reconstruct the entire virus genome from this pool, just like in a classic ARTIC or amplicon type setting. But again, coming back to this concept that we only need one amplicon per primer, this allows us to be really quite free in the way that we design our primers. So rather than just using 72 primers in our pool, some of these primers would sometimes fail. Maybe there's a structural variant or a nucleotide mismatch underneath one of our primers. We could start to throw in essentially hundreds and hundreds of primers. And our final protocol, we had over 335 primers in our reverse transcription mix, each of which would recognize SARS-CoV-2. And these are spaced so closely together that if one fails, it doesn't really matter because there's going to be another one just downstream or just upstream that's going to replace it in its stead. We can use this, we can validate this in the lab. So with Vineet Menachery at UTMB and now at Emory, we showed that we could sequence SARS-CoV-2 from cells in culture. And when we compare random ClickSeq sequencing coverage, around 8% of the reads would map to the virus genome. But when we did Tiled-ClickSeq targeting SARS-CoV-2, over 96% of the reads mapped to the virus genome. So we massively enriched for our target of interest. And that allowed us to look at some of these structural variants that arise during SARS-CoV-2 passaging in the lab. So I mentioned this in this insertion, the furous cleavage site. We could study how deletions in that would be selected or affect the fitness of the virus. And these studies are largely published. And so not only could we do this with cell culture-derived materials, we could also do this from animal studies as well, such as lung turbinates or from tracheal samples and from the nasal lavages in the lab. But these are all experimental settings, which are still quite favorable. So we want to do something more challenging. And so with Dr. Ping Ren and Dr. Scott Weaver at UTMB, we collected hundreds of nasopharyngeal swabs from patients who tested positive for SARS-CoV-2. We could extract the RNA, and we did the precise same Tiled-ClickSeq reaction for SARS-CoV-2. And what we found is that we could get complete genome coverage across the virus genome for each of these patients, as long as the CT value was around 20 to 25, which is a pretty common sort of cutoff for a lot of these studies. Anything higher, we could still capture the virus and get enough sequence information to identify the variant. We just wouldn't get the 100% genome coverage. And so during the early stage of the pandemic, we sequenced hundreds of these, and we could compile multiple data sets corresponding to multiple different variants of SARS-CoV-2 throughout the pandemic. And we published a lot of this work in Jaworski et al. And eLife in 2021, but just very briefly, because we were sensitive to RNA recombination in these patient samples, we could start to measure the differences in RNA recombination between the different variants. So for example, one of the subgenomic messenger RNAs that has a recombination event that gives rise to the spike protein was highly expressed in the B1.2 variants, but was actually much more lowly expressed in what's now called the Delta variant of SARS-CoV-2. And so this was just for SARS-CoV-2. We're now developing these panels. Because we can essentially include as many primers as you want in the reverse transcription reaction, we can start to throw in extra primers. We can look at SARS-CoV-2. We can also simultaneously look at flu or human rhinoviruses, all within the same pot. So with one single reaction, we can actually have a multi-pathogen assay. And then in the lab, we're also looking at, well, why focus just on viruses? Why not look at other transcripts of interest as well that might be rare in a specific sample of interest, but where we also want to be able to resolve features such as alternative splicing or other interesting fusion events or other interesting recombination events that occur in nature. And so just as a very brief example, we've been using Tiled-ClickSeq to sequence messenger RNAs in human samples now. So for example, the protein interleukin 7 receptor, there's both a soluble version that is secreted into the blood as well as a membrane-bound version. And the differences between those two types of the protein is determined by alternative splicing of this exon 6. And work by Mariana Garcia Blanco at UTMB and now University of Virginia, they have shown that this is actually it's correlated as a multiple sclerosis risk factor. And so if you have point mutations in IL-7R as well as an epistatic DDX39B, that can actually be prognostic in your chances of developing multiple sclerosis and the risk of MS over time. And so this exon 6, I think it's this one right here, we designed primers that targeted exon 5 so that we could sequence through exon 6 and map the frequency of mutations in these samples. And so this is just one primer. Remember, we don't have to design multiple primers. If we want to measure the splice event, we don't need to design a pair of primers for every different splice event that might be there. We just have one primer that targets exon 5. And in this assay, we could very directly and very simply measure splice variance. So we could measure the ratio of how many times we saw exon 6 skipping or exon 6 inclusion. And so this is a pretty simple scenario, pretty straightforwardly spliced gene where there's only one intron exclusion. We wanted to challenge ourselves a little bit. So we turned to this protein RBFOX2, which is a master regulator of alternative splicing that also undergoes splicing itself as a messenger RNA. And RBFOX2 has 15 exons in the human genome. And so we designed tiled primers for each of those exons. And just one interesting example, we had primers that recognized exon 5, and when we sequenced upstream from exon 5, we could see that for the most part, we saw splicing into exon 4, but we also sometimes saw splicing into exon 3 as well. So with one primer, we could detect two different types of canonical splicing. In addition to that, we saw non-canonical splicing. So the RNA we used here was the universal human reference RNA from Invitrogen, which contains a lot of messenger RNAs from known cancer cells. We could actually identify cryptic splice sites, so unannotated exon junctions in these sites. Furthermore, from the same primer, we could detect intron retention as well. So our sequence reads, they either map to the junction of the exon and to the next junction, or they read into the intron for giving us a readout of intron retention. So very briefly, from one primer and one assay, we can detect all these different types of splice events, straightforward with very simple bioinformatics pipeline in a simple assay. Okay. So I'm very briefly going to describe polyadenylation, which Eric Wagner will talk about in more detail, so I don't want to belabor the point too much. But in the same way that we can use primers to target a region of interest, we can also use primers to target the poly-A tail of messenger RNAs. And there'll be one poly-A tail for the end of every messenger RNA. And so using an oligo-DHT primer, we can focus our sequencing reads just to the ends of messenger RNAs using that termination. So our azido-nucleotide will only be incorporated just upstream of that poly-A tail. What this means is all our sequence reads will be focused at the 3' end of our messenger RNA. And so Eric will talk more to this in detail. And so I just wanted to go quickly through how we've taken these diverse applications and compiled them into a series of kits that we now market with baseclick. And so overall, what we have is essentially a very modular platform for next generation sequencing. The process is very simple. You don't have to fragment your RNA, and you can use RNA or DNA in this scenario. And your DNA can even be single-stranded DNA. As long as you can prime from your target of interest, you can do a ClickSeq reaction. This gives you a lot of flexibility in how you design your assay, whether it's tiled, whether it's random primed, or it's a mixture of both. You can also start to include unique molecular identifiers or barcodes into your primers in your reverse transcription reaction, for example. And then the click reaction itself is extremely versatile. So you're not locked into a specific platform. You can have oligos at different lengths. Typically when you're using an enzyme, your enzyme will either recognize double-stranded RNA or blunt RNA or double-stranded DNA. With click chemistry, you can essentially click on whatever you like. So you can have a shortened RNA, a long DNA, you can put fluorophores or dyes on it. It provides a lot of flexibility in how you conjugate onto your azido-terminated cDNA. And then we have all of the typical advantages of next generation sequencing. So because we have a single-stranded cDNA that can go to a PCR reaction, we can do all the clever things you would normally do in an NGS reaction. So you can do dual indexing, single indexing. And our libraries, we sequence them on Illumina platforms and Element platforms. And if you take that final library and add on the motor protein and the adapter proteins from Oxford Nanopore Technologies, you can also sequence on Nanopore platforms as well. Finally, this whole process is simple to do. We can do it in 5 to 6 hours in the lab in a fairly standard workday. It's fully automatable. It only uses beads for the cleanups. And so with only 2 hours of hands-on time, you can make a library within a workday easily. The requirements are fairly standard as well. So around 100 nanograms to 1 microgram of RNA or DNA is pretty standard. For the targeted reactions, you can go much lower than that. So for example, when we're doing the Tiled-ClickSeq of coronavirus samples, we didn't even know how much RNA was in our samples. We just extracted everything and put it as an input. The number of PCR cycles you do is typical, 12 to 21. And then the distribution of the fragment sizes in your final library are pretty standard for NGS as well. So you see this nice distribution of read lengths between, say, 200 and 500 nucleotides. And importantly, you can vary that distribution as needed just by changing the ratio of azido-nucleotides you use in your reverse transcription links. So again, we essentially have very flexible control over how we make our libraries. And so all these little modifications and tricks and tips, we've listed them, we've published them, and you can also find them on protocols online at protocols.io. You can leave messages there and comments there. On our website, we have various resources like publicly available data, manuscripts, and what have you. And finally, this allows us to release for general consumption different types of kits, whether you're interested in RNA-seq, DNA-seq, for random prime sequencing, for polyadenylation, or for targeted sequencing as well. And with that, I'll let Eric take over and describe some of the polyadenylation applications.
Okay, thank you, Andrew. I'm going to share my screen here and get my pointer on. All right, there we go. So I want to thank the folks at baseclick and ClickSeq for asking me to talk today. I'm going to sort of give a short presentation from the perspective of a user of this technology. And I'm going to give a sort of a short set of stories focused on one of the projects in my lab for the past 10, 15 years on something called alternative polyadenylation as it pertains to cancer biology. And so I'll give that sort of set of stories, but I'll emphasize my own lab's sort of journey, I guess, or transition from standard RNA sequencing technologies to the ClickSeq that Andrew mentioned. So Nazifa did a nice job giving my sort of my background. I just want to repeat a couple points that I'm a professor in the Department of Biochemistry and Biophysics at U of R. My lab has been studying RNA biology for nearly 20 years. We've published a great deal on that topic, and I'm the Associate Director for the Center for RNA Biology here at Rochester. So to give you all a little bit of background to make this talk a bit more accessible to a diverse audience, I'll sort of refresh you all or maybe even introduce you all to some basic tenets of cleavage and polyadenylation. So shown here is sort of what we envision a pre-messenger RNA to look like, having a 5' cap, having different splicing events, and of course having a poly A tail. And if we zoom in on that 3' end, there are several cis-acting RNA regulatory elements that sort of act together to govern where that poly A tail is to be added. Most notably is the poly A signal, as well as some of these ancillary elements. And together they ultimately dictate where the cleavage will occur. And that RNA then gets cleaved and a poly A polymerase comes in to add a poly A tail. Now these elements exist because there's a machinery in cells called the cleavage and polyadenylation machinery that recognize these elements. And this is a multi-subunit complex containing as many as 20 proteins, perhaps more. And as I mentioned, they recognize these elements and ultimately recruit in an RNA endonuclease called CPSF73 that cleaves the messenger RNA prior to adding the poly A tail. Now, probably for many of you, if you are familiar with cleavage and polyadenylation, I probably equaled your textbook memory of this process. And for most of us, that's all we know because for a long time, the field felt this was a housekeeping event that occurred similarly at all genes. And that sort of put the field to sleep for a while, unfortunately. But that all changed about 15 years ago. And several papers came out. This is one of them from Phil Sharp and Chris Burge's lab, where they took quiescent T cells that were not dividing in one dish. And then in a second dish, they took the same cells that were activated to start proliferating. And they looked at the RNA through microarrays. And they specifically focused on the 3' UTRs. And they made an odd observation that cells tended to create RNAs that were shorter when they underwent proliferation. Now, this was echoed in several other papers at the same time, notably one from Dave Bartel's lab published in Cell. And they found that a similar phenomenon was occurring in cancer cells. So if you look at this northern blot, you can see that the oncogene cyclin D1 in normal breast epithelial cells tended to create an RNA that was long. But in the transformed cells, there was a broad tendency to express a shorter RNA. And so collectively between these papers and many other published at that time, a model emerged where in normal or quiescent or non-cancerous cell lines, there was a general tendency to utilize a poly A signal that was distal to the stop codon. This would create an mRNA with a most lengthened 3' UTR that would basically give the cells maximum control. And that control is elicited through, for example, microRNAs that could target the 3' UTR, of course, RNA binding proteins. And altogether, this would sort of keep mRNAs in check so they wouldn't be overly stable or translate to overly high levels. But in the case of tumor transformation or tumor progression, or even in normal circumstances where cells would proliferate, there's a broad tendency to switch that decision to a proximal poly-A site. And this would have several implications. The simplest, of course, is that microRNAs no longer target this mRNA, so you can get enhanced expression in cis, but it gets more complicated and nuanced than that, because those same microRNAs now could go target other 3' UTRs that perhaps did not undergo shortening, that could actually inhibit their expression. Of course, the RNA binding proteins will now redistribute to other 3' UTRs, so you'll have altered mRNPs. And mRNA localization is also elicited through sequences in the 3' UTRs. So you'll have altered RNA localization. So the bottom line is switching this choice could have profound impact on the cell expression. And one can envision how a tumor would almost be salivating to capture and take advantage of that plasticity. So a little over 10 years ago, that got my lab interested, among many others in the field, to try to figure out how this was occurring. So one of the first papers we published, we did a mini RNAi screen where we knocked down all those members of the cleavage and polyadenylation machinery, and then looked at 3 test genes and looked at their APA using qPCR. And we found that knockdown of one of the subcomplexes called CF1 led to broad 3' UTR shortening of these 3 test genes. And among the 3 subunits, the smallest one, CF25, sort of came in first place. So we decided to focus on that protein CF25, and I want to note it's encoded by the gene, the name NUD21. So I'll kind of use those interchangeably. And we focused on this again because it sort of gave the most robust 3' shortening events. So the main question we had was, what are all the RNAs that are targeted by this RNA binding protein? That's a question for RNA sequencing. And again, if you look at the date, this is going back 10, 12 years ago, sort of in the earlier days of RNA sequencing. So when we did that experiment, I'm not going to belabor these numbers, but for those of you who have done a lot of RNA-seq, you can see that the amount of reads that we did in these experiments was exceedingly high, approaching 300 million, which is, even nowadays, a large number to do for a single sample. And that's because we were interested in the 3' UTR reads, which only accounts for about a fifth or at most a quarter of the total RNA sequencing reads. And so when we did that and mapped those reads, we could easily visualize that 3' UTR shortening was clearly occurring for all 3 of these genes. Now that was exciting to us because we were one of the first labs to see this 3' UTR shortening in RNA-seq, but the problem was we of course knew where to look because we had tested these ahead of time. So to look at all the genes, we had to actually work with another lab led by Wei Li, who's now at University of California, Irvine, and his lab focuses on computational algorithms that sort of are tuned to analyze RNA sequencing data. And we developed a tool called DPARS, which could actually read RNA sequencing data sets, focus on the 3' UTR, and run a linear regression where ultimately it could identify alternative polyadenylation events and quantify them. So we applied DPARS to our RNA-seq, and we found that depleting this CF25 or NUD21 led to a profound shortening in 3' UTRs throughout the whole transcriptome. And many of those mRNAs were more stable as a consequence. When we looked into tumor data sets, we found that glioblastomas, more than any other cancer type, was most sensitive to the expression level of this NUD21, which was almost not surprising because others had learned at the time that of all the tissues in the human body, the brain has by far the longest 3' UTRs of all cell types, and that's conserved all the way down to Drosophila. So these brain cells are probably sensitized to this NUD21 expression. As the gliomas get more aggressive, NUD21 levels go down in expression. Survival is reduced in those patients that have low levels of NUD21. And if we look at those glioblastoma patients, we can easily visualize 3' UTR shortening. And what really sort of nailed it for us was using tumor models where we could express more NUD21 and those tumors grew more slowly, or we could knock it down with shRNA and those tumors grew more aggressively, suggesting that this RNA binding protein is actually acting as a tumor suppressor. So that was sort of the work we had done going back about 10 years, but that generated a key question for us of what might be the critical targets. What are the actual APA events that are most important for glioma cells to drive them through tumorigenesis? Now, we could use standard RNA sequencing. As I mentioned, that worked, and we could use DPARs. But the problem with that approach is it required a high level of reads. As I mentioned, nearly 300 million reads, again, because only a subset of that RNA sequencing is focused on the 3' UTR. And this is where polyadenylation really benefited my research program. Because as Andrew mentioned, this is sort of a focused tool to look at the 3' end of messages. So standard RNA-seq would, of course, have reads throughout the entire gene body or the transcript, which is very useful for things like alternative splicing. But for alternative polyadenylation, we're really just interested in the 3' UTR. So PAC-seq sequencing reads are focused just on that. Now, I'll add through the time period when PAC-seq was developed onwards, we also worked with other groups where we developed actual additional algorithms. In this case, one I want to highlight called PolyA-Miner is a tool that is freely available to all labs in the world through GitHub. But what this tool can do is actually take PAC-seq data set and actually quantify those APA events. And I think what's a really great take-home message for me to leave you with is we were able to run simulations where we actually determined that all you need is in the range of 20 to 25 million reads per sample, and that will give you somewhere around 96, 97% saturation of all the poly site usage in a given human cell type. And I think of equal importance, PAC-seq doesn't just restrict its utility towards 3' end management or monitoring. You can also use PAC-seq to look at differential gene expression. So when we use it, we get basically both sets of information, how genes are being turned up and down, and what is their relative poly site selection genome-wide. And so as an example of this, we published this a couple years ago. Now, again, looking at glioblastoma, LN229 cells are an example of a cell line where we knock down NUD21. We can see, similar to what we saw in HeLa cells and 293 T cells, you get broad 3' UTR shortening. But when we analyze the types of transcripts that underwent that shortening, we could see that they were highly enriched in genes that encoded proteins that are in the extracellular matrix. And so one of those genes, lamin C1, you can see very clearly that in sort of the cells, they have about a 50-50 usage normally, but if we reduce the levels of NUD21, that's almost exclusively at the proximal poly-A site, as are several other oncogenes known to be relevant in GBM, including EGFR, which is measured really throughout the field. And so we sort of focused on LAM-C1, that when we deplete that protein with siRNA or delete NUD21, we could see that not only was there shortening of LAM-C1, but its protein levels were massively upregulated. And when we look at biological outcomes in vitro, for example, migration or invasion of those tumor cells, we see that they are enhanced when NUD21 goes down, again, consistent with it being a tumor suppressor, but that can be rescued if we now RNAi lamin C1. So that suggests that one of the sort of important targets of NUD21 that sort of is driving that metastatic behavior of glioblastoma is at least through LAM-C1. So in the last 3 or 4 minutes, I want to focus on some recent work we have, which is bringing us to the next step. How can we use PAC-seq, study alternative polyadenylation, but now leverage that information to sort of develop cancer therapies? So I want to sort of revisit that set of proteins I was talking about, and I want to focus in on that RNA endonuclease, CPSF73. It turns out that there's actually an inhibitor of that enzyme called JTE607. And it's very interesting how this drug was discovered. It's actually been known for greater than 20 years, and it's known to inhibit AML growth in preclinical models. But for a long time, no one knew what the actual target of this drug was. And actually, it went in clinical trials almost to phase 3. It actually stalled out at phase 2. But it had some effectiveness in these types of cancer cells. So experiments were done ultimately to figure out that unexpectedly JT607, which requires sort of a conversion to an active drug, was able to inhibit an RNA endonuclease, CPSF73, which is encoded by the gene CPSF3. And actually, other labs have solved the structure of JTE607 in the active site for CPSF73. So this was really an unexpected and amazing observation. Up to this point, very few people thought that an inhibitor of an RNA endonuclease in the form of a small molecule could be formed, but indeed that's the case, and it actually could compete RNA from getting into that active site. So now we're working with Mike Fagan's lab at Roswell Park Cancer Institute, looking specifically at pancreatic ductal adenocarcinoma, which is a very lethal form of pancreatic cancer. And we find that CPSF73 is highly expressed in tumor models in his lab. And more broadly, if you look at TCGA, where they have really hundreds of data sets from cancer patients, CPSF73 is more expressed versus normal cells. And looking at cell lines, both in cancer and TCGA data sets, you can see the higher this is expressed, the less there's survival for those patients. And in his lab, the sort of aggressive cancer cell lines have high levels of CPSF73 versus normal epithelial cells, which have very low levels. If we use JT607, at low levels, it doesn't impact these normal cells, but at elevated amounts of these drugs, we can inhibit the growth of these pancreatic cancer cells. And now we're using PAC-seq to find out what might be the critical targets of JT607 that might be beneficial to this type of cancer. And so we did that PAC-seq and we used poly-A minor, and you get now the opposite result, that when you use JT607, you get broad 3' UTR lengthening because you're inhibiting the RNA endonuclease, and that's sort of vividly seen here in these PAC-seq data sets where the drug is causing shifting. This gene is now going from right to left to that distal poly-A site. And when you look at the types of transcripts that are sensitive, many of them are involved in catabolism and autophagy. So we think we're reprogramming these cells in some way that might be beneficial to a patient. And this is ongoing work. In the last two slides, I want to also talk about another project where we leveraged PAC-seq. And this manuscript is now in minor revision at Nature Biomedical Engineering. But we now switch gears to prostate cancer, where we've looked at RNA-seq data sets, combined our DePars algorithm to find that the top RNA that undergoes 3' UTR shortening in castration-resistant prostate cancer encodes a protein called SPSB1, which has been very undercharacterized in the literature. PAC-seq allowed us to map precisely where that proximal poly-A site is, but in this case, we've developed a novel therapy where we've taken advantage of dead Cas13 and used guide RNAs to recruit it in the vicinity of that proximal poly-A site in hopes of forcing those cancer cells to shift usage to the distal site. So we've used a luciferase reporter, where we created a series of guide RNAs near that proximal poly-A site to identify several of them, notably the number 4 and 5, which could reduce the activity of luciferase. It could shift the usage to the distal poly-A site, and most importantly, reduce the expression of this SPSB1 in prostate cancer cells. So in my last sort of data slide here, I'll tell you, as I mentioned, we didn't really know what this SPSB1 did in these cancer cells. So we had to also undergo that line of investigation. We were able to pull this protein down, and it is predicted to be an adapter for E3 ubiquitin ligase. And we found that its main targets are actually the MHC1 complex presenting proteins, which are involved in antigen presentation. And so these are normally deactivated in cancer cells. So we think SPSB1, when it gets lifted, when its expression level goes up in prostate cancer, it is causing immune evasion. And so by adding this guide RNA DCAS13 and switching to the 3' UTR, we can now lower the levels of SPSB1 that reduces the tumor growth in this cancer model. But I think most excitingly, it sort of synergizes with anti-PD-1 checkpoint therapy. And we're currently working through the FDA to try to get this as an approved approach to sort of activate all these immune checkpoint therapies that are currently in use right now, but have limited efficacy, possibly through SPSB1 activation. So with that, I'll sort of end and acknowledge folks in my own lab, and then many of the collaborators that I worked with over the years. Of course, my colleague and friend Andrew Routh, who presented here, through the development of PAC-seq that has really transformed my lab's sort of trajectory and power to study APA. Wei Li and Chan Ben for the work on the prostate cancer and others that I've mentioned throughout the talk. So I'll sort of end my sharing right there and conclude my portion of this webinar.
Thank you very much to all our speakers for their insights and also for their efforts for such interesting research and development essays. So now I'd like to start with our question and answer session. So please feel free and continue posting your questions. I have already received some questions. So the first question is about, I'll ask Dr. Andrew Routh if you can just summarize the advantages of the ClickSeq over the traditional NGS method. If you can summarize the advantages in some short points, please.
Sure. So I'd say there's sort of three main advantages. The first one is the reduction in artifactual recombination. Most NGS approaches that we've looked at generates a lot of artifactual recombination for multiple reasons, such as that adapter-adapter ligation or RNA-RNA, cDNA-cDNA ligation to one another. Because we don't use enzymatic ligases, we get around a lot of those artifactual issues. When we click on our adapter, you can only click on your alkyne labeled sequencing adapter to your azido-terminated cDNA. There's no other ligation that can take place. And so it really brings down the artifactual recombination rate. And because we don't have fragmentation, we can't have that copy choice that occurs during reverse transcription by the reverse transcriptase itself. The second point is that when we click on an adapter, we can include barcodes. So we can have unique molecular identifiers. Each molecule that we click on, each oligo that we click on, that oligo, that one molecule corresponds to the single molecular cDNA that you clicked on. And so computationally, you can control for artifactual recombination, PCR bias, PCR duplication, as well as recombination that might occur during the PCR steps of the library amplification as well. And so then finally, when it comes to targeted sequencing, let's say we're sequencing SARS-CoV-2 or whatever else from a patient sample, if you're using pairs of primers, you're using pairs of primers and like typical amplicon approaches restricts the sorts of recombination events you can find because it relies on those primers being either side of a recombination event at a distance that provides an amplicon that is a similar size to the rest of your amplicons. So it actually places a lot of constraints. So you can still do it, it's just you miss a lot of information. But with Tiled-ClickSeq, because we only have one primer per amplicon, we can sequence essentially whatever is upstream, whether it's a recombination event, different size recombination events, fusion events, you name it. And we've shown this in our clinical studies that this is the case, and this works quite nicely. So I'd say those are the three main advantages. And it's not just us in our lab who's validated this independently. Other groups have used ClickSeq and reanalyzed data and shown that this is the case too.
I'm going to add a comment to that, Andrew, and I totally agree with everything you said. In terms of polyadenylation, another advantage, at least, and I've mentioned it in my talk, but I want to emphasize it even more because it's quite significant, and that is the level of depth needed to get actionable information. With PAC-seq, because of it being focused, we not only get differential expression very readily available in that data set, but we also get poly-A site selection using only around 20 million reads. And where that now becomes so clearly valuable is in the newest Illumina sequencing platforms. And at University of Rochester, we have access to the Nova X Plus, which is the newest machine from Illumina. That their sort of standard flow cell capacity on that is in the range of 1.5 billion reads. So what that translates to is that when we use one flow cell, and at least the Illumina's, I don't want to be quoted on pricing for that, but that's in the $1,000 to $2,000 range. We now barcode on the range of 50 to 60 individual PAC-seq libraries in a single lane. So, just thinking back 10 years ago, that has tremendously transformed just the experimental capacity that one could run. And we collaborate with a lot of labs on using this. And now I find quite often the number one problem I'm telling people is that they need to devise more tests, more experiments, because you could certainly sequence less libraries. There's nothing wrong with that, but you're just getting more reads than you need. So I think, yeah, the depth is a huge advantage, at least for us.
Thank you very much for the answers. There's also a further question regarding to, is there any publication protocol available online where ClickSeq tree azido group was used with ONTs, rapid adapter from rapid barcoding kit, RBT kit? The Tiled-ClickSeq publication only used ligation-based ONT kits.
Yeah, maybe because I answered the first question. That was whether you can actually use the nanopore adapter or this nanopore adapter sequences. However, when you actually want to use the adapter kit itself, that is then you have to do a normal type of ligation. So the enzymatic ligation to already the primers you add via the click reaction. So basically, if you want to use directly the ligation, to the click ligation, to the azido-nucleotides, you have to develop or basically have to get oligos which actually carry an azide group. So meaning you have to synthesize your own nanopore adapters. So you can't use the adapter kit directly from nanopore. That is only possible by a normal enzymatic ligation.
Okay. Thank you very much. And we have also answered lots of questions during the session. So if you have further questions, please don't hesitate to write here. Our experts are there and they can answer your questions. If not, then I'd like to thank all the participants for their interest and for their time joining us today in this webinar. And if there is further question, so please contact us and you can write our experts directly by you'll find also the video of this webinar on YouTube, and or you can write them directly through their emails. And with this, I'll thank you all for joining us today and wish you all the best. Have a nice day. Bye.